So I have titled my talk as Selective Sweep of a SARS-CoV-2 mutant. SARS-CoV-2 mutant is, SARS-CoV-2 is what has made our life so difficult. And I'm going to talk about that selective sweep of a SARS-CoV-2 mutant with some hiccups. And I will introduce the mutant and the kind of hiccups that we are facing. I know that I have 45 minutes, and I will still try to be as clear as possible with as much clarity because of the diversity of the fellowship and of my audience. All right. So I will not make any assumptions, and I'll go ahead, in telling you the concepts and what we've done. First of all, I'm going to run out of time, I'm pretty sure, so I must express my deepest gratitude to these 7 people without whom I would not be able to stand in front of you and give this talk. They not only carried out all of the work, but also provided most of the ideas. And the greatest set of ideas came from this gentleman by name Nidhan Biswas, whose name is, appears bigger than, in bigger fonts than the others. So Nidhan is my colleague. Earlier, he did his PhD with me, but right now my colleague at the National Institute of Biomedical Genomics. And all of these people are with us in our group, and we've done this work together. So I'm very grateful to all of them for not only carrying out the work, but also providing most of the ideas that's embedded in this work. All right, so let me quickly go through the timeline of what's been happening, the unfolding of this particular virus. First, the unfolding happened in very quick time. So to begin with, on December 30th, as a matter of fact, a little before December 30th, this lady who was working in this Wuhan seafood market, she came down with fever and respiratory distress, and she fell sick. But very shortly thereafter, there was a cluster of 27 pneumonia-like cases that were of unknown etiology in the Wuhan. Essentially, most of them were associated with the seafood market. Wuhan is a fantastically large seafood market. So this Huanan seafood market in Wuhan was closed down because the etiology was unknown and people were falling sick. Mostly workers of the seafood market were falling sick. And in January 7th, a novel coronavirus was isolated from the lungs of these people. In 3 days' time, the genome was sequenced. The power of genome sequencing, the technology of genome sequencing has advanced so much that this is a 30,000 nucleotide virus, and the 30,000 nucleotides were sequenced in 3 days. I'm not going to go through each of these, but the first case outside of China was reported on January 13th. So in a spate of about 15 days, it moved up from China to outside of China, and the first case was reported in Thailand. At that time, it was not clear how the virus was getting transmitted, and then it was documented that there was human-to-human transmission, and this was very scary. The WHO declared this outbreak as a public health emergency of international concern on January 30th. So that is within 1 month that WHO came and said that this is of international concern. And on March 11th, the WHO declared the coronavirus disease that appeared in 2019, COVID-19, as a pandemic. Not very often do we face pandemics. The last pandemic that we had faced was in 1918, and that was a long time ago, and that was again related to the lungs and respiratory distress. This particular virus has spread all over the world, as we know, and it's infecting many people. As on 2nd November, 46 million people have been infected globally, with the USA leading the pack, and India is the second in terms of the number of infections. This is not to say that a lot of deaths are being caused by this particular virus. The virus is really a kind virus. It doesn't kill many people, and I'll come back to that a little later, but it is causing a lot of distress. It's causing a lot of infections. It's almost impossible to predict which of these infecteds will actually succumb and die, for whom it will be lethal, and therefore there is a lot of anxiety once you get infected. Also, post-infection, there are a lot of problems that one faces, and many of these infected individuals are actually facing these. But be that as it may, it is continuing to spread, and many people believe that it is going to become endemic to the human society. It will not be eradicated, but it will become endemic, and we have to learn to live with this virus. I don't know about those predictions, and I'm not going to make these predictions. What the observation is that it has spread throughout the world, and the WHO has declared this to be a pandemic. This is so a few things about the virus. Like I said, it has a genome. The genome is an RNA genome, so it's an RNA virus. Our genomes are mostly DNA. This is an RNA virus. There are also DNA viruses whose genomes comprise ATGC. This virus comprises genomes that have nucleotides A, U, G, and C. If you end to end, there are about 30,000 such alphabets. These are called bases, so 30,000 base long nucleotide, 30,000 long RNA sequence is what's embedded within this virus. It's a single-stranded virus, not a double-stranded virus, that is, and it's positive sense, which essentially means that it is almost ready to make proteins. It's like mRNA, it's ready to make proteins. This particular virus has 29 structural and non-structural proteins, or it encodes, the genome encodes 29 structural and non-structural proteins. We are not going to discuss any of those proteins except for one protein, which is called the spike glycoprotein, which is involved in the transmission, infectivity, and the spread of this virus. And we will focus on that. It's called the spike glycoprotein. As you know, that this is a very well-decorated coronavirus, and the flowery things jutting outside is the spike protein essentially, and I will tell you a little bit more about the spike protein because that is attracting a lot of attention with respect to infectivity and with respect to its spread, and therefore with respect to its spread. Where did this coronavirus come from? One way that we inferred this is through nucleotide identity. The more similar the nucleotides are alphabet by alphabet, we suspect that that's the ancestor. So if you look at, and all of these have been sequenced, so there have been coronaviruses that have been isolated from the bat, from the pangolin, and we've had two earlier coronaviruses. There are a few more, but they don't cause any disease. But there are a few more where there were outbreaks. So in SARS-CoV-1, it was just called SARS-CoV. An outbreak had taken place in China, and then the Middle East respiratory syndrome that caused a lot of distress in the Middle East was 10 years later in 2013, and roughly 10 years later in 2019 we have the SARS-CoV-2 coronavirus. The SARS-CoV-2 coronavirus has 96% sequence identity, nucleotide identity with the bat coronavirus, so we think that it came from the bat, or the coronavirus actually came from the bat. But did it come directly? Although the jury is still out, but many of us believe that there was an intermediate host, and the intermediate host was the pangolin. I don't have the time to explain why we believe that the intermediate host was the pangolin, but the pangolin and the SARS-CoV-2, this coronavirus has 91% sequence identity. It's not just sequence identity. There are multiple ways of identifying which may be the ancestral strain and one way or type. The one way we do it is again to do a comparative analysis of the sequences, and what one sees is that the original one, the Wuhan coronavirus, which we believe was the first one to have first individual to have been infected, had in a particular position an amino acid called aspartic acid that's denoted by the letter D, and all of these, the bat and the pangolin, all of them, it's non-polymorphic, meaning that all bat coronaviruses, all pangolin coronaviruses essentially have at that particular position, the 614 position, the same nucleotide, the same amino acid, which is aspartic acid or aspartate. So this again bolsters the view that this coronavirus SARS-CoV-2 probably descended from the bat via the intermediate host called pangolin. The viruses cannot survive without their hosts, and we are interested in this virus not only because it's infecting us, but why is it infecting us? The first thing we need to recognize is that viruses cannot survive without their hosts. They need a host in order for their own survival. Because they don't have the appropriate cellular machinery to be able to replicate, and they hijack some of the host cellular machinery, such as when they infect us, they will take some of our cellular machinery and use it to their advantage in order to replicate. So they have to enter the host cells, otherwise they are dead, so to say. Therefore, because they use the host cells in order to replicate, the viruses really don't want to kill their hosts. And like I said, I've used the word kind for the SARS-CoV-2. SARS-CoV-2's killing spree is less than 3 individuals of the 100 individuals it infects. And there is also a great age disparity. It's primarily most of the deaths have occurred in the elderly people, or in people who are compromised with respect to their lungs, or they have diabetes or blood pressure. So there are certain comorbid conditions where you have a greater killing rate, or the virus's killing rate is greater than the younger people in the population. So anyway, it's killing less than 3 people per 100 people it's infecting. But the other viruses, the previous coronaviruses, were not so kind. The SARS-CoV-2, that was an outbreak in Wuhan. It didn't even pan out as an endemic. It killed about 11 people of the 100 people of each of the 100 people that it infected. And the Middle East Respiratory Syndrome coronavirus, that was more lethal. It killed about a third of the people that the virus infected. But be that as it may, viruses cannot live without their hosts, and it is therefore not to the virus's advantage to kill their hosts. So they are usually quite kind. They are spreading, they infect very well, and we need to understand the infection. And that's the reason why one does these kinds of studies. These various viruses have preference for certain cell types. So we are very familiar with the hepatitis viruses. They are all mostly infect the liver. This year's Nobel Prize went for discovery and research on hepatitis C that homes into the liver, so that resides in the liver. The SARS-CoV-2, this coronavirus, has preference of host cell types. It prefers the lungs, and there's a reason why it prefers the lungs, and I'll tell you in a minute why it prefers the lungs. But it also infects other places, such as the gut of the human, the nasopharyngeal, the airway ducts in the human body. And those are the preferred places where this particular coronavirus likes to inhabit and likes to infect. Those are the cells that it likes to infect. The reason why it likes to infect those cells is because there are 2 things that a virus must do, or at least this coronavirus is doing. It needs to attach to the host cell, and then having attached to the host cell, it needs to enter the host cell. Because unless it enters the host cell, it cannot hijack the cellular machinery for its own advantage in replication. So it attaches itself to a protein that's on the surface of the human host cell. In this particular case, we know that the spike protein actually attaches, and it attaches itself to a protein called angiotensin converting enzyme. Many of you may be familiar with this. This is also something that's associated with human blood pressure. But angiotensin converting enzyme is expressed on the surface of these lung cells, gut cells, and so on in plenty, and therefore the more the merrier for the virus, because it can find a lot of attachment sites, and once it attaches, the probability that it will be able to enter the cell also increases. The more the number of viruses that can attach to a specific cell of the host, the probability increases of its entry into the host cell, and therefore its success in replication. To be able to enter the host cell, the viral membrane and the host cell membrane must fuse. So there is a fusion element associated with it, and this fusion element also makes a hole in the cell wall, so to say, in the host host cell wall, through which the virus then enters the host cell, and now it starts hijacking the machinery. So it's infected the host once it enters the host cell. So I said that there are essentially two components to this entry. One is called the attachment, where it attaches itself, and the second is fusion. So this is a cartoon that actually explains all of this. So it moves from the attachment to the fusion, and then to the entry of the virus into the host cell. But cartoon apart, what's important is that I talked to you about the spike protein. The spike protein has essentially two components, two subunits, so to say. One is called S1 and the other is named as S2. Both of these subunits, each of these subunits is actually specialized to do a function. The S1 subunit is specialized to attach itself to the host protein, which in this particular case is angiotensin converting enzyme 2. And the S2 subunit is supposed to be able to allow the viral cell membrane and the host cell membrane to fuse with each other. The two subunits are joined at some point of time in the spike protein, and these two must be separated. They must be cleaved for them to start their function. So the S1 and S2 must be cleaved, and there is a place where there is a joining of the S1 and S2, and this joint must be cut so that S1 and S2 are separated, and then they can start doing their usual function, which is attachment and then fusion. This cut is also done by a host protein, and I will come to that in a minute. So essentially, there are multiple stages that are involved. So this is again something that tells you a little bit more about this process. One is that it is attaching itself. There is this ACE2 receptor. It attaches itself. This is the angiotensin converting enzyme, the protein to which, the host protein to which the virus attaches itself. And then there is a protease. A protease is again a protein that actually cuts the cellular membrane, and this is called TMPRSS2. This is a specialized protein that's called a protease, serine protease, that actually cuts the cellular membrane, and then the virus is then able to enter. So essentially, there are two players, and you will see a third player in a minute. So the two players are angiotensin converting enzyme to which it binds, to which it attaches, the virus attaches, and then the other host protein called TMPRSS2, which actually helps cut the cell membrane or fuse the cell membrane. Okay, so I'll come back to this in a little later. But we, those of us who are interested in sequences and genomes of various kinds of various organisms, we have been facilitated by two fantastically rich global databases. One's called GIS-AID, and the other and its curated database called NextStrain, which is really nice because they have been compiling influenza and influenza-like viruses and sequencing them and compiling their and collating their sequences. These sequences allow us to estimate various things. For example, we've been now able to estimate the rate of mutation, rate of accumulation of change in the genome of the coronavirus, and the estimate is about 24 changes, 24 nucleotide changes per year. But per year for a virus, as you know, that the generation time is really very, very short, days, sometimes hours, and therefore 24 substitutions, 24 changes per year is really a very small mutation rate, very, very small mutation rate. The other thing that I must point out about this particular coronavirus, many of the RNA viruses do not have the proofreading machinery, meaning when it replicates, then errors come in, errors build up, and there is a proofreading machinery that actually proofreads whether there have been mistakes in copying. Most RNA viruses do not have that proofreading machinery, but this particular coronavirus, SARS-CoV-2, has proofreading machinery, and therefore their rate of errors during this copying process is small compared to many of the other RNA viruses. So we've been able to estimate the rate, and what we have done, our group has done, essentially is to use these sequences that are available in this particular database to our advantage to answer several kinds of questions. And we have actually early in May, so this paper was published in May in the Indian Journal of Medical Research, and we have analyzed RNA sequences at that time About 3,500 SARS-CoV-2 sequences were available in this next strain or GISAID database, and they were collected from 55 countries, sequenced, and the sequence was available. So we had analyzed these, and we proposed, or we discovered, that there was a selective sweep of one particular virus type, and this virus was a mutant type. It was not the original type that arose in Wuhan, probably descended from the back by the pangolin, but a later mutant, a changed virus that was sweeping throughout the world. And that discovery we made, and we published this in the Indian Journal of Medical Research. This was in May 2020. Subsequent to our publication, there were two quick publications, one from Iceland in the New England Journal of Medicine, which was in June, and another in Cell, which was in July. So in quick succession, three groups independently discovered that one particular virus type was sweeping, and the paper in Cell actually did some cell biology studies to show that this particular mutation that I'm going to talk to you about at length actually provides greater infectivity, is actually more infective than the original Wuhan type that, again, I will talk to you about in a minute. So what we've done is that we looked at these genomic characteristics, and I'm not going to describe all of the genomic characteristics, but eventually what we were able to do is figure out that there were, aside from the original type that was called O, and these are called clades, that was called O, this O type was the Wuhan type that's ancestral. Subsequent to that, that particular ancestral type accumulated changes in its RNA sequence, and these changes were of such nature that they led to different branches of a phylogenetic tree. And the phylogenetic tree is a very fast evolving phylogenetic tree, because the generation time, if I can call it the generation time of a virus, the generation time is really short for this particular virus, or for any virus for that matter. We were able to identify 10 more subtypes of this original type. So in all, there were 11. There still are 11. Well, there may be one or two more smaller branches that are evolving, but by and large, there are these 11. Of these, 9 of these, including the original type, has at a particular position, in the 614 position, an amino acid called aspartate, and two of the other evolved subtypes that are named as A2 and A2A, they have at the same position, at the 614th position, a nucleotide change that results in glycine, an amino acid called glycine instead of aspartate. So we are talking about a nucleotide change that leads to an amino acid alteration from, at position 614, from aspartate or aspartic acid to glycine. And this particular mutant that I'll be talking about is the 614G mutant, glycine mutant, is what is actually sweeping throughout the world. Remember, the timelines have been very, very short in terms of its first detection to its spread, first to Thailand and then to other parts of the world. And we have been following this virus, so to say, or sequences of this virus, and this is what we found on 31st of March. Remember, the WHO declared the SARS-CoV-2 as a pandemic on March 24th, and this is on 31st of March. 31st of March, this particular evolved type, the A2A type, which is not the original, the evolved type, has actually swept most parts of the world, and about 50% of all of the sequences that have been, all of the viruses that were sequenced belongs to this particular subtype A2A. So this particular subtype must be enjoying some kind of an advantage, because of which it's able to sweep through. It's gaining a selective advantage and is able to sweep through the world. As the days proceeded, this particular subtype, the A2A subtype, or the 614G glycine subtype that comprises A2 and A2A, started sweeping through the world even more. And as we can see, by early May, this particular, the larger branch of this phylogenetic tree is the A2A. And if you combine A2 and A2A, both of whom have the 614 glycine, and the right-hand side tree explains the green is the original, the aspartic acid, and the orange is, or the golden color is the 614G, which is the glycine mutant. As you can see, that By early May, it's almost 70%. By July, this is what's happening. By July, it's about 80%, maybe 85%. So it's essentially sweeping through the world. So it had gained selective advantage. I will tell you a little bit of the cell biology experiments that were done, but the Cell paper that I talked to you, which was published in July, that already provided some hint that this particular mutant, the glycine mutant, has some advantage in order to be able to infect human cells better. They already had provided. Subsequently, there have been many more studies, and that has been bolstered that it does have a greater infectivity, or it gains infectivity. It's able to infect human host cells better, the mutant. What we observed, and this is again July. These two graphs pertain. The top one pertains to Europe. The second one pertains to North America. The blue, or the light blue, the sky blue, pertains to the original type, which is the aspartate at position 614, and the red is the mutant type, which is glycine at position 614. As you can see, by July, both in the United States and in Europe, the Caucasian population, so to say, have been completely infested or infected by, not the population, but whoever was infected with the 614 mutant type, which is a glycine type. So this is fantastic. It has a selective advantage. It has swept through all of these regions, the countries in Europe and North America. When we looked at the data a little bit more closely, other parts of the world, what we found is that in East Asia, there have been hiccups. The virus is facing some hiccups in East Asia. It's not able to spread itself so well. As you can see from that graph, it's nowhere close to 100%, and there are periodicities in terms of the original subtype versus the mutant subtype. So it is facing some hiccups. So this was interesting. Why is it that it is able to spread through the Caucasian population so well and is not able to spread through the non-Caucasian populations? Can there be something in the host genome that is preventing the virus from spreading in non-Caucasian populations? So we asked this question, and the first thought that came to our mind, or we also looked at that data, did some modeling of that particular data. This is the modeling that we did. And Europe and North America, they were asymptotic almost at 100%. East Asia might become 100%, but at this time it's much lower. It's about 77%, and we don't know where the asymptote is going to be. Probably it will be 100%, but it's going to take a much longer time compared to North America and Europe. So the question that we are asking is, or we had asked is, we believe that we have an answer. That's why I'm giving this talk in the first place. So why is the question that we asked is, why is the coronavirus finding it so difficult to sweep through non-Caucasian populations in East Asia? Just to recall, I mentioned that the spike protein latches on to this protein called angiotensin converting enzyme, which is on the cell surface of these lung cells, and it needs to latch on because it needs to gain an entry. If you look at this ACE protein, which is a human protein, if you look at that gene is long. It has 26 different exons or protein coding, polypeptide coding regions, and anyway, it has multiple changes. So it's polymorphic. There are multiple polymorphisms. There are variants at various positions in this particular gene. Some of these variants are benign, meaning that it's non-amino acid changing. It does not have any impact on the level of production of this particular protein. But there are some, for example, in the promoter region, and most importantly, this insertion deletion polymorphism that does change some properties of this protein. And so what we thought is that probably the frequencies of the variant alleles at these particular positions are different between the Caucasians and the non-Caucasians, and that's probably the reason why the virus is probably the lung cells of the non-Caucasians are not producing as much ACE protein, and therefore not as many viruses are able to latch on to the lung cells of the non-Caucasians, and therefore they're unable to infect them so well. So we looked at human databases. Again, there are human variant databases that we are aware of. We searched all of the databases. There are multiple. We searched all of the databases, and this is what I'm not going to show you the data, but this is the conclusion that we came to. Like I said, that there are some variants that impact on ACE expression levels, and we believe that the frequencies of these variants may have been higher in the non-Caucasian populations than in the Caucasian populations, and therefore it was not expressing as much protein in the lungs of the non-Caucasians, and therefore the virus was unable to gain entry. But what we found to our dismay is that this was not the explanation, because there were no significant difference between Caucasians and non-Caucasian populations in the frequencies of these variants that actually impact on ACE expression levels. We also did that for TMPRSS2, and we couldn't find. TMPRSS2 is the other protein. So the way that we reasoned is that first it's unable to latch on, maybe, but that was not it. Then we reasoned for ourselves, is it possible that the serine protease, the TMPRSS2, has certain variants that are present in differential frequencies between Caucasians and non-Caucasians. In the non-Caucasian population, it's unable to cut that, cleave that particular cell membrane in a meaningful way or as efficiently as in the non-Caucasians, but even that we had to sort of discard. That was so ACE and TMPRSS are not the major reasons why this is happening, why the coronavirus is finding it difficult to sweep through non-Caucasian populations compared to the Caucasian populations. We then, so like I said, that the TMPRSS2 is the other protein. So we reckon that the original type, the aspartate acid containing type at the 614 position, and the mutant glycine, both of them have the appropriate cleavage sites or machinery for cleaving. And therefore, so far as cleaving is concerned, they are equally efficient, the mutant and the original, the ancestral type. However, what we discovered, and this was our discovery when we did bioinformatic analysis and other kinds of analysis, what we discovered is that the mutant, the glycine mutant at 614, actually creates an additional cleavage site, and the cleavage is important because the virus needs to gain entry, and this we discovered that they had the D2G mutation, which is the variant mutant type that we are talking about, creates an additional cleavage site, and this additional cleavage site is a neutrophil elastase 2 cleavage site. So we said that it's quite possible that because of this additional cleavage site, which is only possessed by the mutant virus and not by the ancestral virus, this mutant virus is able to gain entry into the host cells more effectively. So this is a neutrophil elastase. So we thought that maybe the Caucasians have greater numbers of neutrophils circulating in the blood, and the non-Caucasians probably don't have as many neutrophils, and therefore the neutrophil elastase availability is greater in the non-Caucasians, in the Caucasians than in the non-Caucasians. We looked, and what we found was that, I'll come back to this in a minute, what we found was that there was no evidence that there was a higher count of neutrophils in the non-Caucasians, in the Caucasians than in the non-Caucasians. So there was no significant difference, and this was a paper that was published some years ago in the American Journal of Human Genetics, and what we found is that with respect to neutrophils count, there was no significant difference. We also, this was a hypothesis that the creation of the additional cleavage site will enable the virus to will enable the mutant virus to be more infective. This was actually done by a group where they took human cells and infected them with these two different kinds of viruses, the original and the mutant. And what they found was that there was a fourfold more efficient entry of the virus of the mutant virus than the original virus, and that's the reason why we believe that maybe neutrophils are important. But what we found was that at least in terms of number of neutrophils, that was not the simple explanation by which we could explain this Caucasian non-Caucasian difference. Then we were also aware that there is an inhibitor of neutrophil elastase, and that inhibitor is called alpha-1 antitrypsin. Alpha-1 antitrypsin inhibits neutrophil elastase, and so we started reasoning whether alpha-1 antitrypsin may play a role. Alpha-1 antitrypsin, we know that there are some individuals who have normal levels and some individuals who are deficient in alpha-1 antitrypsin. And alpha-1 antitrypsin is coded by this gene called SERPINA1, which is located on chromosome 14. This particular gene also, like all other human genes, also has certain kinds of mutations, and mutations in specific mutations in SERPINA1 causes alpha-1 antitrypsin deficiency. There are many alleles, there are many different variants in SERPINA1 that impacts on the level of alpha-1 antitrypsin, but there are three main alleles: the M allele, the S allele, and the Z allele. The M allele produces normal levels of the alpha-1 antitrypsin protein. The S allele produces moderately lower levels, and the Z allele produces very low levels or no alpha-1 antitrypsin at all. So if you look at the genotypes, the MM genotypes have normal levels. The MS and MZ genotypes have slightly deficient levels of alpha-1 antitrypsin, but the SZ and the ZZ genotypes are deficient in alpha-1 antitrypsin. And please remember that alpha-1 antitrypsin inhibits neutrophil elastase. So if you have a lower level if you are deficient in alpha-1 antitrypsin level, there will be a lower inhibition of neutrophil elastase. That will imply a lower inhibition of neutrophil elastase, and if it's inhibited lower, the level of neutrophil elastase is going to be higher. So neutrophil elastase actually provides a cut site or a cleavage site, so there will be greater level of spike protein activation of the SARS-CoV-2, and there will be a higher level of infectivity because spike proteins are now activated more. There is greater cleavage, and there will be better spread in population. So we said that if this logic is correct, then, and if this logic is an explanatory logic for the mutant virus to spread so well, then lower levels of alpha-1 antitrypsin must promote better spread on the population. So that's what we then looked at. We looked at East Asia, Europe, and North America. The reds are the mutant, where it spread very well, and East Asia, where the reds are lower and the blues are higher, blues or greens are higher. The greens are the non 614Gs, and the reds are the mutant 614G. The blue ones are the ancestral type D614. If you look at this, lower inhibition of neutrophil elastase means better spread in the population, and this is the deficiency, and this is in Europe and this is in North America. What we find is that there is higher deficiency levels of neutrophil elastase and lower deficiency levels of neutrophil elastase in East Asia, and that correlates very well with the spread of this particular virus, the mutant virus 614G. We looked at it, the correlation even at the country level, and if you look at countries in East Asia and if you look at countries in Europe and North America, as we can see, the correlation is much higher. It is very good even at the country level, not just at the region level, and therefore what we believe is that we have an explanation as to why this particular virus has been able to spread so well in Europe and North America, and it is because that there are greater number of people with alpha-1 antitrypsin deficiency among the Caucasians. The non-Caucasians have lower alpha-1 antitrypsin deficiency, and therefore the virus is not able to spread so well. So deficiency of alpha-1 antitrypsin is actually promoting the spread of the virus. The virus is able to gain its selective advantage, meaning it's able to infect better in individuals who are deficient in alpha-1 antitrypsin. So we came up with a model. Where the right side is the individuals with alpha-1 antitrypsin deficiency. The left side, the left lung, is individuals with normal levels of alpha-1 antitrypsin deficiency. And again, I don't want to leave room for a few questions, so I don't want to explain this in great detail, but essentially this is a summary of what we have said in terms of the neutrophil elastase and its relationship with alpha-1 antitrypsin, and how it increases the efficiency of infectivity of the mutant virus, which in turn is able to spread itself much better in non-Caucasian Caucasian populations than in non-Caucasian populations. Thank you very much for your attention.